Hello and welcome to the Sheekey Science Show, where in this video I had great fun speaking with Professor Gordon Lithgow and one of his PhD students, Edward Anderton, discussing all about the worms C.elegans and small molecules that prolong lifespan through enhancing molecular stability. I hope you enjoy our conversation. So, hi Gordon, hi Edward, thank you both for joining me today and welcome to the Sheekey Science Show.
Thank you for the invitation, it's great to be here.
Yeah, it's great.
No problem. So I thought I'd start with one of the kind of generic questions, but a question that's always interesting to hear different people's perspectives, which is, what is aging to you and how do you think we should be describing aging? As I know you've been using the concept and talking about the aging field in general, but Geroscience as well as a concept.
Yeah, I mean, it's a huge question actually, really, isn't it? And I think we're still not very good at actually defining aging, to be honest. We know how to manipulate it. And we can talk about all these pathways and tissue changes and molecular changes, and we can do all of that. There's textbooks of that. But the question, what is aging, is still, I think, elusive in a way. To me, it still is this deterioration with time. It's an increase in mortality, age-specific mortality rates, which is a very terrible definition, but at least it's something you can say that's true. That happens in almost all animals. But the Geroscience thing, and I want you to pile in here, Edward, I mean, the Geroscience thing is really the idea that whatever aging is, if we at some time have a chemical biology explanation of aging, whatever it is, we do think it causes disease, chronic disease in late life. And it's everything, all the cancers, Alzheimer's, Parkinson's, osteoarthritis, osteoporosis, the big list of diseases. And the key there is the word cause, because that's not generally, I think, still accepted by the wider biomedical community, the idea that aging is a cause of something. And I don't know, I mean, when you started thinking about aging, did you think about it as a cause?
Of disease?
Yeah.
Yeah, I mean, it seemed kind of obvious from the research, because we can manipulate the aging process, right? So we have interventions that can slow down the aging process, or at least it seems that way. And usually there's a concomitant decrease in the prevalence of disease, or disease is pushed away, which would suggest that there's a causal aspect to it. I don't really have anything to add in terms of a definition of aging. I think you did a really great job there.
Yeah, but I mean, it's worth noting that you are of maybe the first generation of younger scientists who kind of accept that idea of aging as a cause of disease. If you go to all of the major neuroscience institutes and cancer institutes across the world, you'll find very little expertise on aging itself. And that's kind of stunning. If aging is really the cause of these things, how is it that we're as a biomedical community are still not employing that in our ways to find new drugs and new interventions? Yeah.
So I mean, how are you using the word Geroscience then to kind of communicate what this field really is?
Hopefully, the word was, we came up with the word here actually at the Buck Institute, and it was 15 years ago, and it was to represent the fact that we had expertise in disease, and then people working on worms and flies and yeast, and suddenly realizing we're all working on the same thing. And we thought, well, what is this? It's not biology of aging, it's not geriatric medicine. And so we came up with an umbrella term, Geroscience, to try and describe the interface between the sort of normal aging processes that we're studying and the disease processes.
I see. And you brought up worms, so C.elegans is how I kind of know them, but that's kind of like, I guess, your model organism of choice. So I guess, can you talk more about why and what's so fascinating about these creatures as a model organism?
Yeah, I mean, they're a fantastic model to study aging precisely because they have a very short lifespan. So they only live approximately 2 to 3 weeks in the lab. We understand the genetics quite well. We understand the development quite well. They have a set number of cells, and we understand how those cells develop. And what's interesting is that in many ways, the C.elegans is a good model for more complex aging in mammals and in humans, because they display many of the same aging phenotypes. So they slow down with age. They get a sort of sarcopenia where the muscles degrade, their intestinal lumen degrades, and so they get a kind of leaky gut, which is seen in mammals too. And so in many ways, you can study these diseases of aging in the worm over a very short period of time.
I mean, it's truly remarkable, just to repeat what you've said in some way, so the first mutations that were shown to extend lifespan in the metazoan were found in C.elegans back in Tom Johnson's lab in 1988 in University of Colorado. And you know, I got interested in that at the time, but I never imagined we'd be having conversations about human health around these same genes. And then more recently, we've seen, you know, sort of chemical compound-like or drug-like interventions. And of the published and unpublished stuff that I know about, it's looking like you can go from the worm to a mouse at some sort of hit rate like 50%, which is unbelievable, really. I mean, to think about it, so we're really hitting conserved biology. So you go back and say, well, actually, the worm has proven to be amazing, that we can actually go into a mouse in sort of preclinical setting, whether that be aging pathologies or lifespan itself, and see effects from these compounds that were identified in worms. I'm stunned by that.
Yeah, I mean, I think there was a paper that you published a couple of years ago where you've identified about 6 different compounds that seem to be able to extend the lifespan, and that's in like the C.elegans, like equivalent of the mouse ITP interventions testing program.
Oh yes,
that's a similar C.elegans worm-based program. And so could you elaborate more on this and talk about potentially these different compounds that seem like most promising?
Sure. I mean, the program itself was, it came about because some of us felt there was a problem with the scientific literature so we published a paper, flashy paper in Science, you know, antioxidants extend lifespan. And then 2 other labs, excellent labs, could not repeat that data. And it was really shocking at the time. And we kind of moved away from even using chemical compounds in the worm at all for a while. And it was only a decade later that people started to say, well, this is actually happening too often. It's happened more than once. We could always replicate our own data, but we simply couldn't tell other people how to do it, you know. So the National Institute on Aging set up, first of all, a mouse intervention testing program, then a worm intervention testing program. And the idea was to say, you know, sort this out, work out what's going on, you know, find protocols that work between labs. And essentially, both organizations have done that. So we published our first paper testing a bunch of compounds that were kind of probably well known to some people. They'd been in the literature already in different ways. But we established a protocol where all 3 labs, and this is Patrick Phillips's lab and Monica Driscoll's lab across the US, all 3 labs could replicate the data. And they did not see variation between the laboratories themselves. And so some of the compounds that came out, thioflavin T was an example of an amyloid binding compound that worked in, and it didn't just work in the lab strain. This is a critical point, actually. It wasn't just the lab strains. We also tested wild strains, recently brought in from the wild, and different species of Caenorhabditis. And Caenorhabditis is incredibly genetically diverse. And so what you're really doing is you're testing the idea that if a compound works in this genetic background, could it also work in a different one? And that's relevant to us, right? That's relevant to humans. And so yeah, it was kind of interesting. So we did find a couple of compounds that really worked well. We found a whole lot of others, including at least one that we'd published previously did not work at all. And then, but you know, maybe one of the biggest things that came out of this was the variability in experiments and the realization that we don't document enough about how we're doing experiments in order for people to replicate.
True.
And actually just the sort of natural variation in lifespan experiments from run to run was enormous. It was amazing. We had no idea that existed until we did this kind of thing at scale.
Yeah, and it's interesting you brought up thioflavin T, which I know, yeah, as you say, binds like amyloid fibrils, which are kind of like aggregated proteins. And so does these compounds that you have arisen from these tests, has that therefore given us some indication of the like underpinning molecular processes?
Well, yeah, I'll hand it over to you to talk about proteostasis, but you probably realise that every aging scientist you talk to thinks that they're working on the most important mechanism of aging. So we think this is the most important mechanism of aging. But just describe where we are with this.
So your question is, how have we learned from the compounds, or how have we learned about the role of protein homeostasis in aging?
I guess
Both. Yeah, so maybe your best place to talk about THIO-T and what we learned from those studies, because that was prior to my time in the lab.
Yeah.
But in terms of protein homeostasis, I mean, my thesis project revolves around a project that started in the lab a decade ago now, really, which was looking at how we know that with aging, this isn't just in worms, this is in mammals too, there's an increase in these detergent insoluble protein aggregates. So that's just protein aggregates which stuck together and become these very resistant protein aggregates. And so what they found is that with aging, this happens in the worm, and we can extract these protein aggregates and we can look at them by mass spectrometry, so we can see what kinds of proteins aggregate with aging. We find that it's very reproducible. It's the same kinds of candidate proteins that come up all the time. So what we found is that there are certain compounds, to kind of answer the question, that can reduce the levels of this the insoluble proteins. So, for example, vitamin D. If you feed the worms vitamin D from an early age, it massively reduces the amount of protein aggregates that you see with aging. And you see this massive lifespan increase too. But it's not a simple story, actually, because it seems like maybe there are some protein aggregates that are actually beneficial. So it might be the case that protein aggregates have evolved to kind of soak up toxic monomers. So there are some people who think that might be the case in Alzheimer's disease, where you see these plaques. So there are compounds that we think, like thioflavin T, where, contrary to our prior belief about with the vitamin D story, they actually increase the level of protein, insoluble proteins. And so it could be the case that thioflavin T and other amyloid binding proteins are protected because they kind of accelerate this process in a beneficial way. So they bring down these monomers into protective aggregates.
And one thing we like to do is combine pharmacological approaches with genetics as a way to try and get at mechanism. And with thioflavin T, we went into it thinking, thioflavin T binds aggregating proteins, therefore it might prevent aggregation, which it does in a test tube, and therefore that might be a great thing. And it worked. But then when we started doing the genetics, we realized it wasn't just THIO-T itself, it needed all the components of proteostasis in the worm to actually work. So if you took away chaperones, if you took away the heat shock factor, if you took away the proteasome functions or autophagy, it didn't work. And so there's this dependency on natural protein aggregation degradation processes to see the beneficial effects. And maybe one other thing to say about this is that the proteostasis, I think, makes a really important connection to disease, because as Eddie described there, that the detergent insoluble proteins, they're behaving biochemically like A beta, alpha synuclein. The proteins of very significant, terrible neurological disease, exactly the same. And we believe that that's not an accident. There's a connection there that's important.
So I guess on from that, then obviously Alzheimer's is something that we've been struggling to try and cure for a long time. And so therefore, how translatable do you think that the discovery of these different compounds will be to human diseases?
I mean, we're looking at it. We're actually moving compounds from worms through into mammalian cells, through into mouse models. It's hard to say. I mean, I think that many of us, at least in the aging community, think that one of the major failures in Alzheimer's therapy development has been ignoring the aging component. And in many ways, some researchers have been using mice that have an accelerated Alzheimer's pathology, almost eliminating the aging component at all. So then you cure that, then you go into a real, animal or person, and the aging component is the dominant thing that's causing the disease. So I don't know. I would love to think that this is a really novel and possibly successful way to develop therapeutics for Alzheimer's. And the NIA have put significant resources into, I think, testing this general idea, which is great. So we'll see.
Yes, I mean, it's pretty promising, especially. And so there's something you kind of hinted at a bit earlier with going back to this ITP testing study, where you said that you saw variants even within this one species with the same intervention. And so, I mean, I think I saw something once about how this same thing with the C.elegans study, Nicholas Strauss-Style.
Mm-hmm.
I might be pronouncing it wrong, but he showed that there maybe was like the stochastic element to aging, or some noise where like everything, the conditions were the same, they were genetically identical.
Exactly.
So what's happening in this situation?
It's bizarre actually, and people have been doing these experiments for, I don't know, 25 years or something, and for the first time we saw, because we did so many of them, that you would set up an experiment under a very tight protocol, very standard operating procedure, and you get a result. Worms lived 15 days. You set it up again, same strain, not even with a drug at this point, same strain, and now the worms are living 22 days on average. And we would do this again and again and again, and in the end, actually, what we saw was that the lifespan was kind of biphasic. There was something we call it stochastic. Obviously, it's something we're doing, but we don't know what it is. But in all three of the labs involved in the CITP, we would have short lifespan and long lifespan for the same strain under identical conditions.
Interesting.
And so, in a way, it made me feel better, because I thought, well, this maybe explains some of the problems in the literature. Because one lab could be in a sort of short-lived mode, another lab could be in a long-lived mode. And then you come in with a drug, and who knows what's going to happen under those different physiologies or biology or something. And it's the kind of thing where, I don't know, in the past you would see it like a short lifespan, and you would think, oh, well, something went wrong with that experiment. There must be contamination or something else going on there. And you would just throw it away. And we decided, no, nothing gets thrown away. Everything is real. Everything is natural variation on this particular phenotype of lifespan. And I'm at peace.
And so speaking just of how you do these experiments, how do you actually go about studying C.elegans? Because I mean, I'm sure to many of my viewers that seems like a crazy, how do you study worms? What's involved?
Yeah, I mean, obviously that's a really simple experiment you just described, where you can just add a compound to a plate and then you can watch the worms die, essentially. You can score them when they die. Sounds gross. But
we poke them with a wire,
we poke them to see,
and if they move,
yeah,
they're alive.
Yeah.
That's about as sophisticated as it gets sometimes.
Yeah, we can't check their pulse. So I mean, but they're actually transparent, which is amazing because it means that you can look at the body structures down a microscope. And so there are many different labs that generate these amazing strains where they express proteins with like a GFP or RFP fluorescent protein, and you can just see it. I mean, because the worms are transparent, so you can watch trafficking of proteins through the autophagy pathway, for example. Milena Hansen's lab, who they're new to the book, and they just moved up here, and they're like the world-leading experts on this. So they have these fantastic strains where you can look at the quenching of these fluorescent these fluorophores as they move through. That's just one example.
And then the transparency. Any student can come into the lab, or a high school student come into the lab over the summer, show them young worms and old worms, and they're on agar plates, or there's a little bit of E. coli on there for food. And it's blindingly obvious which are which. The young worms are moving around pretty fast, they're eating, they're moving sinusoidally like this. The older worms get to the point where they're not moving at all, and you literally look through the skin, and you see that the tissues are completely disorganized, and there's all sorts of weird vacuoles and dark patches and things. It's like aging is happening in front of your eyes for 20 days. And I think that's something that many of us continue to be attracted to the worm model, is that when you see a mutation or a drug that stops that from happening, there's a switch goes off in your head. You go, this is incredible, that this idea of lifespan is malleable, it's plastic, it can do something about it.
Yeah, exactly. And I think that goes back to what we were saying earlier about how we define, and how, I guess, to both of us, aging as a cause of disease did seem kind of obvious based on all this exciting data. And so we've spoken about some of your research, but what are the main, I guess, research questions in your lab?
I kind of leave it up to these guys, to be honest. We as lab leaders, we all have our own ideas, but the real creativity comes from the trainees, the students, and the postdocs, and so that means that things change all the time. So, for example, a couple of years ago, one of our colleagues, Julie Anderson, she had a PhD student who was very interested in exercise and Parkinson's disease, and but then sort of wanted to do some worm work on this, and so there's an exercise model for the worms. So she worked with her lab, Mina Schmidt, published a really nice paper on exercise, and then another PhD student comes to the lab wants to work on exercise as well. I had nothing to do with any of that, right? The general themes are really about the relationship between stress, as shown by the proteostasis example that Eddie was talking about, this lack of protein homeostasis with age, and then now, much to my surprise, the connection with disease. And we definitely have shifted from genetics to a more chemical biology approach. So we're very interested in continuing to screen for compounds that suppress aging pathologies in the worm. Like you can make a worm that exhibits aspects of Alzheimer's disease or aspects of Parkinson's and so on. And those worms are super easy to screen for chemical compounds that suppress those phenotypes. So a lot of the compounds we've been talking about up until now started back in those screens like vitamin D. We had no intention of working on vitamin D. That wasn't an idea. That came out of a screen of natural products that suppressed Alzheimer's pathology in a worm. And then we realized that actually had these amazing effects on normal aging as well in the worm. And surprisingly, that was super interesting to clinicians because they don't really have a good explanation as to why vitamin D deficiency in adults leads to elevation in risk of disease, cancers, diabetes, neurological disease. And we looked at that and went, that's aging. That's accelerated aging. So human deficiency could be, in some way related to, again, a hypothesis, not a conclusion, but yeah. So I think that the themes move around all the time, and that's great because every new person that comes into the lab brings new ideas. But the overall theme is, can we explain this sort of aging disease connection?
So firstly, how do you actually exercise a worm? But secondly, I mean, just what are the limitations as well with working with this model organism?
Well, what do you think, the limitations?
Maybe I can answer the exercise first. Yeah, so really simply, the worms will naturally swim when you put them in water or put them in a buffer solution. So you can simply pick up the worms and drop them in solution and they'll swim happily for a few hours. And so they do these characteristic body bends.
Nice.
Yeah.
and they become exhausted. So they'll only do it for a certain amount of time, and then you can tell that that's their limit. Kind of thing.
And it makes them hungry actually. So once you put them back, because obviously they're not in their food at that time, but when you put them back to their food, then they'll gobble it all up much faster than they usually do. So, kind of like humans.
Sorry, what was the second part of the question?
Limitations of working with worms.
Yeah, I mean the limit. Well, when you submit a grant, everyone tells you about the limitations, and it's a worm, right? It's not a mouse, and there's a lot of biology that must be specific to the worm. But going back to the amazing success rate of moving from worms to mice with chemical compounds, I think says that it's despite the limitations, it's a good model to find things that would be important for human aging. So one example is our colleague Simon Mello published a paper we were collaborators with last year, where he tested a compound that we'd identified in the worm as extending lifespan. And he was looking at it in the mouse, and he was doing a whole panel of different things like cardiovascular health, metabolism, bone health. And he showed that this compound was reducing the rate of bone loss by about 30%. So a very significant change in bone loss. And then our colleague Julie Anderson has preliminary data showing that this self-same compound is protected against Parkinson's disease by protecting the dopaminergic neurons and preventing alpha-synuclein aggregation. So you've got this compound that you find in a worm that doesn't have bone, that barely has a brain, and then you move it into a mouse and it protects against both domains of aging. And I think that's amazing. I don't know if this is going to be common. I mean, obviously rapamycin is another great example of this.
Yeah.
But it does say that maybe this sort of geroscience idea has got something going for it, in that when you're going down a drug development route for Alzheimer's, you're possibly also going down a drug development route for other chronic diseases at the same time.
Oh yeah, for sure. I mean, so is this work published? What is the molecule you want
The bone work is published. Yes, it's in a bone journal.
So what was the molecule that was found?
The molecule is called HBX, and actually it's structurally related to thiaflavin T, which we were talking about earlier. We chose HBX because it's a bit more drug-like. We actually did not see a lifespan extension in the mouse, but most of the mouse studies, you're looking at one concentration. You're making a best guess. They're very expensive. You can't do a whole, I mean, this entire study had 700 mice in it, but with 4 interventions. So it was a large study. But even then, you can't. It's hard to go back and say, well, let's try another 10 concentrations and see if we can get the lifespan effect. But the effect on bone was amazing.
And so I guess we've spoken now about the limitations of the worm, but what's the future potential of the worm in terms of how is new technology being incorporated? So we have CRISPR, new high throughput methods of screening, imaging techniques, but how is that being applied to studying worms?
I mean, all of it's been applied. I mean, CRISPR's obviously making it easier to make strains and to go into these wild strains that are recently isolated, so you're not dealing with sort of lab adaptive strains. Single cell is now possible in the worm, and that's such thing. Wow. Yeah. It's just I mean, any technique you hear about, someone's going to try it in a worm.
Yeah.
And I think you always want to use the best possible system to ask the question you're asking, for sure. And many times that's not the worm. And we've got people in the lab who are working in cell culture, and obviously the mouse work that we've done. For me personally, I think it's the aging component that Eddie talked about earlier, the fact that this worm ages so rapidly. There's results within 20 days for the most part. And I keep coming back to that thinking, well, if we're interested in combinations of compounds, if we're interested in compounds that have much larger effects than we've seen, exploring new chemical space, we really don't explore much chemical space. Really, I mean, there's only been tens of thousands of compounds looked at for aging, mostly in worms. And there are millions and millions of compounds, as you know, and there's different chemistries that create really interesting compounds that are very different from the drug-like libraries. And when I look at that, I think, yeah, we'd probably just do it in a worm.
Yes, I mean, how much space do they take up in terms of storage space?
Oh, well, they're 1 millimeter in length.
Yeah.
We grow them on agar plates that are yay size, and we keep them in plastic box in incubators at 20 degrees C, which is optimal for reproduction. And I mean, compared to the mouse, my goodness, this is an incredibly economical model, right?
Yeah.
Although it's still labor intensive, a lot of things are still done manually, despite some automation that we use.
Okay. And they're hermaphrodites.
They are.
So what does that mean for sex-dependent differences?
That's
also something I'm interested in at the moment.
Yeah, that's a really good question. I mean, you can generate males, and so it's possible to do males versus hermaphrodites. And that's something that actually the lab has just been talking about. It's in the lab meeting this week. We're thinking about maybe screening some compounds in males to get around some limitations of working with hermaphrodites. But yeah, I'm not sure how much of a hermaphrodite is a female. I don't know if we can make that distinction.
Yeah. I mean, there's certainly lots of mutants where they're truly female or
right.
Generate males all the time, that kind of thing. So there are variations there to think about. There isn't enough done on this. It's a really, really good question. And actually, I think the worm is a very interesting model to think about these things. And other people like Jennifer Garrison here at the Buck are certainly thinking about this kind of approach. And historically, as you know, it's been a disaster. I mean, most mouse research has been done on males alone.
Yeah.
And thankfully, the NIH has kind of fixed that, and it's impossible now to have a grant on males only. That's a great thing. So.
Yeah, I mean, I guess it will be interesting to see if there are differences when you compare mice and hermaphrodites.
Yeah.
And so, I guess talking more generally about the fields where it is at the moment and its future potential, are there any other areas of longevity research that you're interested in? There's been a lot of interest around cellular reprogramming at the moment. So has that been tried in the worm? I don't know. I mean, just what are your general interests besides proteostasis and worms?
Yeah. That's a lot.
Yeah. So this is something that we've talked about quite a lot in the lab about can we do reprogramming in worms? And it is the case that they do have orthologs of the Yamanaka factors, and so it is, in theory, it should be quite a simple experiment to do. And I know that I'm pretty sure there's a lab in Switzerland that's trying to do that right now. So but we haven't tried that, so I can't say anything about the data right now.
Yeah. I mean, this institute has something like 160 scientists working on aging or age-related disease, And so it kind of represents quite a lot of diverse approaches. You know, metabolism, epigenetics, you know, you can imagine we have a huge interest in cellular senescence here, which, you know, has been amazing to watch because 20 years ago, it was not even obvious that cell senescence actually contributed to aging. And even the best people in the field, Judy Campesi, to name one here, you know, would say, I don't know if this is really contributing to aging. But my goodness, it's amazing how it's developed and how it's grown. And so that's like, I think everyone's interested in that, even if we're just spectators, right?
Yeah. I would say I'm biased because that's what I study, but yeah, they're definitely very interesting. And so I think more just like generally now, like, do you have any advice for people being like scientific advice, like how the due diligence in performing experiments, or just like more general advice if you're curious and finding out more information about science?
Oh, that's good. I mean, I think that it's really important to read broadly if you can. I don't right now, but the people in my lab do, and you can just see all these ideas percolating up to the surface. And collaborations. I think collaboration is possibly the most important thing we've got at the moment. You know, between labs in a single institute, between universities, between institutes across the world. You know, and so Eddie's collaborating with Max Planck right now, and I think that's super important. And it's hard. It's hard to make these things work. But stick at it, you know, talk to people. When we get back to going to meetings, go to meetings, you know, and make connections with people and you know, come up with experiments that work for both of you or all three of you or all four of you. I think that's really
important. Yeah. I'd like to say a bit more about that. I think that everybody maybe at some point in their life has had this feeling of imposter syndrome.
For sure.
Yeah, it's still happening. And I think people should just realize that, you know, everybody's been there, and that, you know, everybody's trying to learn, and that reaching out to somebody and saying you think their research is cool, can we have a chat about it, is actually seen as a positive thing. If somebody reach out to me to have a chat about my research, if anything, I'm flattered by it. So don't be afraid to reach out to people you've never met before.
At any level. Scientists love talking about science, right? And they love talking about their own science, especially. Yeah. So once you make that move, I don't think anyone ever says, "Nah, I don't want to talk about it." You know, I mean, they want to talk. Yeah.
And I've definitely been realizing more and more that it's going down an academic path doesn't mean you have to stay in academia forever. And so, you know, the biotech space right now is exploding in aging, and I mean, in the Bay Area, it's just crazy how many people are interested. And I don't think that it's once you leave academia, you can never come back either. I think that the doors open in both directions.
Right, absolutely.
So I would encourage people to just think broadly about it and, you know, keep their options open.
Sure. And so where do you see like the field in 10 years' time? I know that's a hard question, but yeah, do you have any thoughts?
It's always 10 years, isn't it?
Yeah,
I mean, I'm very optimistic for the field. I think we will see breakthroughs in humans in the sense of suppressing human aging pathologies and maybe disease. But the field has to deliver that, and you know, all the biotech interest right now is phenomenal. I love it. I'd love to see this because it's money coming into a field that was essentially. It was a science project, right? That's what it was. It was basic scientists doing basic stuff, following their curiosity and loving doing what they're doing and publishing their papers, and that was it. But then there was this rising hope that, well, this is actually could be important for people. And so to see an industry emerge is really, really exciting. And I dearly hope that we'll get some wins from this in humans in the short term, and then I think it will explode. And not to be too dramatic, maybe within 10 years, biomedicine will be transformed by this field. That's what I hope. I'm optimistic.
Obviously, I'm in the same position as you. I feel optimistic about it.
You're hoping to have a job in 10 years.
Yeah, I mean, obviously, the results of the TAME trial should be out in 5-6 years. Mm-hmm. And so maybe metformin will be the first aging drug. I wouldn't be surprised if we see wins with rapamycin in human trials. Yeah, I feel like you said, it's the floodgates may open at that point, because if people realise that we can attack all of these different age-related diseases in one go, then everyone's mind will be open to that geroscience idea.
And it's you guys that are going to do it. It's your generation, and the very fact that you're here, communicating to people about this field is super exciting. Thank you for doing that. And I think that the more we can inform people that there's something potentially special could happen that could transform maybe even their own lives in their late life, it's truly amazing. I mean, one of the problems I think the field has had is the challenge of talking about aging, especially for people who are a little bit older. You can't just take a camera through a geriatric psychiatric ward and see the misery there and come out the other end and say, do something about this, people. People don't react to that very well. It's too hard. It's so hard. And it's almost impossible to imagine we can do something about that. But what's happening in the animals says we can. So that's where the optimism comes from, I guess.
Yeah, for sure. I mean, the same as you both, I'm also optimistic, and I hope that, can also contribute to the field. So I just want to say thank you to both of you for talking about your research. It's been a really interesting and fun conversation. So I'm sure we'll hear more about the worms.
Absolutely. They're not going away.
Yeah, sure. So thank you again. And yes, it's been great fun.
Thank you very much.
So I hope you've learned something in this video. Thank you to my Patreon supporters, and thank you for listening. So hi Gordon, hi Ed. Sorry.
You can just call me Edward, it's fine.